Assalamualaikum warahmatullahi wabarakatuh. Our topic is about Atropine eye drop, and it will be presented by me, Muhammad Sharifuddin Ansari bin Muhammad Nasrum, and Muhammad Hanif Naim bin Zainuddin. For the introduction, Atropine eye drop are a medication used primarily in ophthalmology for their ability to dilate the pupil, which is mydriasis, and to treat conditions such as uveitis, amblyopia, which is lazy eye, and to aid in eye examination. The active ingredient of the atropine sulfate is an anticholinergic drug that works by blocking the action of the acetylcholine on the muscarinic receptor in the eye, leading to pupil dilation and reduced accommodation. It is also utilized to relieve pain and inflammation associated with eye condition. Typically available in 1% solution, atropine eye drops are prescribed by the healthcare professional and should be used with caution, as overuse can cause adverse effects like increased intraocular pressure or sensitivity to light. So next, what is a certificate of analysis? A certificate of analysis, or COA, is a document issued by the Quality Assurance or Quality Control Department that confirms the product specification and quality characteristic of a particular batch of goods. The purpose of certificate of analysis is provide detailed information about the composition, property, and test result of the product. This information help ensure that the product meets the required quality standard and specification. So this is certificate of analysis of the Atropine eye drop. It contains certificate number, product code, product name, who is supplier, manufacturing batch number, seed cost lab batch number, date of manufacture, and expiry date. It also have list of tests that have been testing to the Atropine eye drop product. So firstly is certificate number. The certificate number assigned to this product is 5. It is essential to maintain a direct correlation between the number provided on the product label and the unique identification number printed on the certificate to ensure traceability and authenticity. Next, we have the product code. The product code for Atropine eye drop is NDA 206289. This code assigned by the manufacturer or regulatory authority serves as unique identifier for tracking and referencing the product in official document, order, and quality assurance. Each line item subject to FDA regulation must have a product code that aligns with the product name or invoice description to ensure clarity and compliance. Next, supplier. The supplier of the Atropine eye drop is DKSH Malaysia Sdn Bhd, known for its expertise in market expansion service. DKSH ensures the effective distribution and availability of the product across various regions and markets, maintaining accessibility for healthcare provider and consumer. Next is manufacturer and batch number. The batch number is 307002 is assigned to this specific production of the Atropine eye drop. This number is a critical reference that enables the tracing of the manufacturing history, ensuring transparency and quality assurance throughout the product life cycle. Next, product name. The product name officially named is Atropine sulfate ophthalmic solution USP 1%. As a pharmaceutical product, it carries multiple designations: the chemical or scientific name, the generic or non-proprietary name, and the brand name commonly recognized by the public. This designation ensures precise identification and usage in clinical and regulatory context. Next, date of manufacture. The Atropine eye drop were manufactured on 2011-2024. This date signifies the completion of key processing steps, including the addition of the active pharmaceutical ingredient to other components, ensuring the formulation meets required standard. Next is expired date. The expired date for the Atropine eye drop is 27/12/2025. When stored under recommendation condition, the product remains stable and retains its strength, quality, and purity until this date, ensuring its effectiveness and safety for use during this period.
Next is the standard operating procedures for atropine eye drops, which is atropine sulfate ophthalmic solutions. For our products, there are several tests that will be conducted to ensure the test meets the quality, safety, and regulatory compliance standards adhering to the guidelines such as USP guidelines. Firstly, we begin with the appearance test, which confirms the solution is clear, colorless, and free of visible particles. The solution is inspected visually against both white and black backgrounds under sufficient lighting. Any cloudiness, discoloration, or particulates result in failed batch. These tests adhere to the USP 771 for ophthalmic products and USP 790 for visible particulates. The second one is the assay test. Determines the concentration of atropine sulfate in the solution. Using high performance liquid chromatography HPLC, the sample is diluted with dilute hydrochloric acid and injected into the HPLC system with a UV detector set at 254 nanometer. The concentration is calculated based on the calibration curve created with atropine sulfate standards. This test complies with the USP 621 for chromatography and USP 771. Next is the pH test. The pH test ensures the solution pH is between 3.5 and 8.5 around that value of the pH. Which is suitable for ophthalmic applications. The pH meter is calibrated using standard buffer at pH 4.0 and 7.0, and the solution's pH is measured at room temperature around 20 to 25 degrees Celsius. This test follows the USP 791 and USP 771. The specific gravity test. It measures the solution density relative to the water. A clean pygnometer is weighed empty, filled with the solution, and weighed again. Specific gravity is calculated as the weight of the solution divided by the weight of the water at 25 degrees Celsius. This test adheres to the USP 841. For the solutions containing benzyl conium chloride as a preservative, the preservative content test ensures the correct concentrations. The sample is diluted with a suitable solvent such as methanol and analyzed using HPLC with a C18 column and UV detection. The preservative level is calculated based on the calibration curve of known standards. This test complies with USP 1225 and USP 771. For the sterility test, it ensures the product is free from microbial contamination. Using the membrane filtration method, the solution is filtered through a sterile of 0.5 micromembrane in an aseptic environment. The membrane is incubated in fluid thioglycolate medium, which is around 30 to 25 degrees Celsius, and soybean casein digest medium, which is around 20 to 25 for 14 days. Microbial growth indicates a failed batch. This test then follows USP 71 for sterility testing. The endotoxin test is conducted using the Limulus amebocyte lysate, which is LAL, kinetic chromogenic assay to ensure the endotoxin levels are below 0.1 European per milliliter. The sample is mixed with LAL reagent in a microplate, and the adsorbance changes at 405 nanometer, which is measured to determine the endotoxin levels. This test adheres to the USP 85. Next, the microbial test ensures the microbial counts are within the acceptable limit. The solution is filtered through a sterile 0.45 micromembranes, and the filtrate is cultured on trithic soy agar (TSA) for total aerobic microbial count and suborate distors agar (SDA) for yeast and malt count. TSA is incubated at 30 to 35 degrees Celsius, while SDA is incubated at 20 to 25 degrees Celsius. Colony counts are compared to the USP limits, and these tests follow USP 61 and USP 71. The viscosity test ensures the solution consistency and ease of application. A Brookfield viscometer is used to measure the viscosity at 25 degrees Celsius. The viscometer is calibrated with standard fluids before the test. This test complies with USP 912 for viscosity. The clarity and color test checks for solution's transparency and the absence of discolorations. The solution is visually inspected against white and black backgrounds and under the proper lighting. Optionally, a turbidimeter can be used to quantify turbidity. This test adheres to USP 631 for color and achronicity and USP 771 for the clarity. Finally, the packaging test ensures that the containers are intact, leak-proof, and properly labeled. Leakage tests involve pressurizing or vacuum sealing containers, while labels are inspected for the accuracy, including the critical details such as the batch number, expiry date, and storage conditions. This test complies with the USP 6C1 for containers and USP 771 for the ophthalmic packaging. For the summary, Atropine eye drop, which is a 1% atropine sulfate ophthalmic solution, is used for pupil dilations, and the eye exams, which is rigorously tested to ensure the safety and efficacy. These SOPs guarantees the compliance with the USP standards with the Certificate of Analysis (COA) that we have been constructed, which provides the critical details such as the batch number, product code, and manufacturing and expiry dates. Together, the SOP and the COA can uphold the product quality, regulatory compliance, and the patient safety. That's all from us. Thank you.